Hi everyone, am I audible, visible? Hi everyone, am I audible, visible? Hello. Am I audible, visible? Please let me know in the chat section. So happy morning, everyone. Welcome to Unacademy NEET English. I'm your biology educator, Ambika Sharma, and today we are going to start a new chapter, that is sexual reproduction in flowering plants, right? So you know that in the morning sessions, we are covering class 12 syllabus. In the evening, we are covering class 11 syllabus, right? So I hope you guys are preparing your notes. I hope you guys are revising your completed chapters from the NCERT. And yes, do practice the MCQs as well. Okay, do practice the MCQs as well, because that are also very important. MCQ practice is the most important thing. And do you know, bache, sometimes we want to know the ways by which we can revise, we can cover our more syllabus in less time, isn't it? Isn't it? Hannah? Sometimes this is what we need. We need the trick so that we can revise our more syllabus in less time. So this is what I'm going to do today. Today I will post one video, right, in which I will tell you about the tricks, the ways by which you can revise a syllabus, right, by which you guys can revise more syllabus in less time. Okay? Done. So now let's start the topic and let's start the chapter sexual reproduction in flowering plants. But before that, let me tell you, students, see here you guys can see our channel. If you are new to our channel, first of all, subscribe our channel, because it is one of the best channels for the NEET preparation. Here we are providing you the quality academic content, and moreover all the important information related to your NEET. Whatever you need to know about the NEET examination, you will get it here. You can see here, time to time teachers are providing you the mind maps, right, how to revise NCERT. It is a very important video, do check it out. Okay, and moreover, right, the short videos for revising a topic, every important update you guys are going to get here. So join, subscribe to this particular channel first of all, and if you want to be the part of our batches, and especially the mind maps, right, it is started by HSP sir, and let me tell you, students, right, it is one of the best batches for your chemistry. Okay, so what you have to do, just a minute, here you people can see in the description box there is a link. Just go to that link, click there and be the part of this batch. Okay, now let's start the class. Let's start our chapter, that is sexual reproduction in flowering plants. It's a very easy chapter. First of all, so when it comes to this particular chapter, so in this chapter, the microsporogenesis, megasporogenesis, apomixis, pollination, these are the most important topics, and the embryo sac, these are the most important topics. From this particular chapter, 3 questions can come in your NEET examination, and they are mostly the repeated ones, right? Let me tell you, they are mostly the repeated ones. From this particular chapter, you are not going to get anything difficult. Okay, you're not going to get anything difficult from this particular chapter, because it is actually a very easy chapter, right? Here we are going to talk about the flower first, about all the parts of the flower and how the gamete formation is taking place there, right? Like first of all, microsporogenesis, then the development of male gametophyte, megasporogenesis, then the development of female gametophyte, right? The pollination we are going to discuss, right? So these are the most important topics there. Okay, seed, the difference between albuminous and ex-albuminous seeds, these are the most important topics of this particular chapter. So time to time I will tell you that yes, this question is the repeated one, that question is the repeated one. That's what you need to do. So this lecture is going to be more than sufficient for your NEET examination. Clear, bache? Right? Okay, so now let's start this particular chapter. So the first thing is sexual reproduction in flowering plants. So means we are talking about the angiosperms here, isn't it? We are saying that sexual reproduction in flowering plants. And now you know that in the plant kingdom, right, your angiosperms are the flowering plant. Yes or no? Your angiosperms are the flowering plants. We are going to discuss this chapter in a very different way, in a very unique way. We will revise it. We will relate it with the plant kingdom as well, okay, for your better revision of plant kingdom as well. So when we talk about the plant kingdom, so you know that we'll be discussing algae, bryophytes, pteridophytes, gymnosperms, and the angiosperms, isn't it? That's what we discuss in the plant kingdom. Now, when you talk about the algae, they are very primitive type of plants, okay? They are photosynthetic. They are important. I'm not saying they are not important, but they are primitive. In their case, there is no proper differentiation of root, stem, and leaves. Such things are there. But again, it is very important. Then the amphibians of plant kingdom. Now, when you talk about the pteridophytes, the gymnosperms, and the angiosperms, when you talk about the pteridophytes, gymnosperms, and the angiosperms, do you know they are the tracheophytes? For them, we use the word tracheophytes. And do you know the meaning of word tracheophytes? They are the tracheophytes. What are they? Pteridophytes, gymnosperms, angiosperms. They are the tracheophytes. The meaning of tracheophyte is that they have vascular tissue, right? They are the first plants. Basically, pteridophyte is the first plant where you will see the vascular tissue. That is your xylem and phloem. Are you getting it? That is your xylem and phloem. Are you getting it? Right? And then after pteridophytes, you will see gymnosperms and angiosperms. So gymnosperms and angiosperms, they are the plants having seeds. Okay, they are the plants having seeds. So we also call them as phanerogams. Okay, and spermatophytes. When it comes to the gymnosperms and the angiosperms, you call them as phanerogams. You call them as spermatophytes. Fine, you call them as spermatophytes. Okay, so yes, you are right, Subhashini. Gymnosperms and angiosperms, they are the seed-bearing plants. What are they? They are the seed-bearing plants. So that is why we use the word phanerogams. We also call them as spermatophytes. These things are very important for the revision. Now, students, in the case of the gymnosperms and the angiosperms, if you talk about their life cycle, so it is diplontic life cycle. Okay, no doubt we have to start with the flower, but before that you should know about the angiosperms. Fine. So which type of life cycle is there? It is the diplontic life cycle. Which life cycle is there? It is the diplontic life cycle. Now, what is the meaning of being diplontic? Right? What is the meaning of diplontic life cycle here? Let me tell you. Now here, it is the sporophyte. It is the diploid phase, sporophyte, which is the dominant one, right? It is the long-lasting one. It is the photosynthetic one. Students, when we are saying it's diplontic life cycle, means majority part of the life cycle is having the diploid phase, right? There, the plant is having the diploid phase, and for that we use the word sporophyte, the structure which is going to form the spores. Clear, bache? The structure which is going to form the spores, right? So sporophyte is the dominant photosynthetic and the independent one. Okay, and the independent one. And when you talk about the gametophyte, okay, so it's few cell stage, it's single or few cell stage, and it is dependent on sporophyte. Is it clear? It is dependent on sporophyte. That is the meaning of diplontic life cycle, right? I have posted one short video on these life cycles as well. If you want, you guys can check that too. Clear? This is the meaning here. This is about the angiosperms. So quickly, if I have to tell you about the angiosperms, they are the flowering plants. Okay, they are the most advanced plants. They have the xylem and phloem. They have the seeds, and here in angiosperms, seeds are covered. Okay, they are not naked, they are covered. So the main body is the dominant part is the sporophyte, the diploid phase, the photosynthetic phase, and the independent one. And when you talk about the gametophyte, it is single or few cell stage and it is dependent upon the sporophyte. So up to this part all clear? Yasir, Trisha? Yes? Up to this part all clear? Now, next is flower. Next is what? Next is flower. Okay, so sexual reproduction in flowering plants, that is our angiosperms. Now, what is this flower? Actually, this flower is a modified shoot. What is it? It's a modified shoot. Shoot is forming the flower. And here, flower, it's the seat of sexual reproduction. It's the place where the sexual reproduction will take place. Fine. These are the two most important points about this flower. What is it? It is a modified shoot. Okay. And second is, it is the seat, it is the site of the sexual reproduction. It is the place where the sexual reproduction will take place. These are the 2 points. Now, the third one. Just a minute. Okay. Okay. So these are the 2 important points. Now, what is happening? See, we know that plants will continue their growth, right? For the lifetime in the plants, the growth will be there, right? In the plants, when you talk about the growth, it is for the lifetime. Why? Because in the plants, we have the meristematic tissue. What type of tissue is there? There is a meristematic tissue. Now, when the word is meristematic tissue, it means that this is the tissue having actively dividing cells. Okay, so in meristematic tissue, we will see small size cells which will keep on dividing, right, bache? And they are responsible for the growth. So here in meristematic tissue, you know that you have the apical meristematic tissue, right? You have the lateral meristematic tissue. That is a different thing. But mainly, why am I telling you this? That when it comes to the meristematic tissue, it is present in the apical region, means at the tip region. Okay, at the tip region. So you have shoot apical meristem, you have root apical meristem. The meristematic tissue which is present at the shoot tip is the shoot apical meristem. The meristematic tissue which is present at the root tip, it is the root apical meristem, right? It is the root apical meristem. Now, when you talk about the flower, as I said, it's a modified shoot. Okay, what is it? It's a modified shoot. So actually this shoot apical meristem, okay, it will form floral meristem, because ultimately the flower will form from a shoot. So that's what I'm saying, that this shoot apical meristem, it is going to form the floral meristem. It is the one which will form floral meristem, and this floral meristem will form your flower. Okay. Okay, so let's revise it. So shoot apical meristem, it will form floral meristem, and this floral meristem will form the flower. So that is what you need to understand. And yes, bache, floral meristem will completely be utilized. Like in the case of shoot apical meristem, it will keep on dividing. It will help in the primary growth of that particular plant. But when you are talking about the floral meristem, it will be used entirely. It will be used for the formation of a flower. So what is a flower? Flower is the site of the sexual reproduction. That's what you need to keep in your mind. So up to this part, all clear? If there is doubt, do let me know. And you know that this part is related with the anatomy of the flowering plants, and it is not even difficult, let me tell you. Okay, it is not even difficult. Clear, bache? Clear, bache? So as per, yeah, so this is what flower is. Now, as I said, it's a modified shoot. So now when you talk about the flower, first of all, let's talk about its parts. So you know that there is many times there is a stalk-like structure, isn't it? Many times there is a stalk-like structure. Let's discuss about its parts first. You know that there is a stalk, right? This stalk is known as pedicel. What is it? It is pedicel. Now this flat dome-shaped structure, it is known as thalamus. Again, it is also a modified shoot. It is also known as receptacle. Thalamus and receptacle it is. Now, here, what will you see? See. That's the female part and the male part, right? That's the female part and the male part plus. See this. Like this diagram is explaining all. I know it's not so beautiful, but it will explain all the parts here. Okay, so just have a look. So as I said, the stalk-like structure is pedicel. This flat dome-shaped structure is thalamus or receptacle. Again, it is also the modified shoot. Now, when you look at this part, right, this part, it is basically the sepal. What's that? It is basically the sepal. Now here, these are the petals. These are the petals. Now this part is your anther, and the stalk-like structure is again your filament, right? Anther and filament. So together, this anther and filament, they are known as, it is known as stamen. Fine. See, pedicel, flat, thalamus or receptacle, these two, these are the sepals, right? Here you have the petals. This is anther plus filament. Together they are making the stamen. And this part here is the pistil, and we also call it as carpel. What is it? It is pistil. We also call it as carpel. Clear, bache? This is what a flower is. A modified shoot and having these parts. Okay, it's a modified shoot and having these parts basically. Clear, bache? Clear, bache? So here you can see the stalk, pedicel. So its free end is having this thalamus and receptacle. Again, in the bracket here you can write down that it is the modified stem, right? It is the modified stem, right? These are the parts here. Now we have to see which one is essential, which one is non-essential, right? And how are they useful? This is what we are going to discuss next. Done? Let's start it. See, in the case of flower, because it is a modified shoot. Now, in general, you know that when you talk about the stem, how do we differentiate a stem from the root? When you talk about the stem, it contains nodes and internodes. Yes or no? I hope you know about it. It contains nodes and internodes. When you talk about a stem, it contains nodes and internodes, right? And on that basis, we differentiate it from the root as well. That always in the case of stems, even if you will see any underground stem modification, you will find roots, nodes and internodes. But such structures are absent in the case of roots. Okay? Such structures, they are absent in the case of, yes, bache, they are absent in the case of roots. Clear, bache? Clear, clear, clear. So, flower is also a modified shoot. So, of course, it will also contain nodes and internodes. Like here you have the thalamus, which is again a modified stem. So this thalamus is going to bear nodes and internodes. Because flower is a shoot, that's why flower is also having nodes and internodes. It's just that nodes are placed very close to each other, right? Internodes are not very long here, right? Nodes and internodes, they are placed very close to each other, right? So you can say that internodes are small and highly reduced. So let me mention this. So when you talk about the flower, as I said, it's a modified shoot. So it contains nodes and internodes. Okay, nodes and internodes are highly reduced. Highly reduced here. Clear, bache? Internodes are what? Internodes are highly reduced here, right? So these nodes, for these nodes, we also use the word whorls, right? We will also call them as whorls. That flower is having the whorls. Clear? So these whorls are nothing. They are what? They are the nodes basically. Clear? These whorls, they are nothing. They are just the, yes. Right, bache? Kind of nodes there. So actually, right, if I have to be more precise, these whorls are kind of modified leaves which are present on thalamus. Clear? These are the modified leaves which are present on the thalamus. These whorls basically. Okay? Modified leaves which are present on the thalamus, the flat or the dome-shaped structure also known as receptacle. Okay? also known as receptacle. So they are also the modified leaves or the modifications of the leaf. So we have 4 nodes, right? In a flower, in a complete flower, what will you see? You will see four nodes or 4 whorls. Clear? 4 nodes or 4 whorls. So the first one is calyx, denoted by K. Second one is corolla, denoted by C. Then androecium, denoted by A. And then gynoecium, denoted by G. Right, it is denoted by G. So calyx, corolla, androecium, gynoecium. Calyx, corolla, androecium, gynoecium. See, bache, here you have the sepals, right? Sepals. Can you see them? They also look like some kind of leaf-like structures now, right? The best flower to understand it is your rose, right? Even your rose, even your China rose, right? If you will look at the China rose and the rose, you will learn about all these things. Like let's start with the rose. So in the case of rose, you know that when it is in the bud stage, right, that green colored structures, they are covering that particular bud. Yes or no? That green colored, some leaf like structures, they are covering that green bud. Actually, they are the sepals, right? They are meant for the protection. They are protecting that growing bud. Right, so their main role is of what? It is of protection. Are you getting it? Sepals. So the group of sepals, it is forming the calyx. What is calyx? Calyx is denoted by K. It is the group of sepals. Many sepals together. For them, we will use the word calyx. We will use the word calyx. Now, these are the petals. Red colored petals in a red rose. Right. They are what is their main role? To attract the insects for the pollination, right? What is their main role? They are very brightly colored. In different flowers, these petals are brightly colored. They have beautiful colors. Why? To attract the insects for the pollination. Are you getting my point? For the pollination and to attract the insects. So here, the group of that petals, okay, group of petals, it will form corolla, and it is having the different shapes. In chapter morphology, we have, in the chapter morphology, we will study it. Okay. Now the next is androecium, right? See, anther plus filament, stamen. See, stamen. In the spelling itself, men is given. So it is the male part. It is the male part. So this androecium, A, denoted A. So it is basically the group of stamen. Okay, it is basically the group of stamen. And gynoecium, G for gyne, G, girl. So it is the group of, basically you can say that here what are we going to discuss? Here we will be having the pistil or carpel, right? Pistil or carpels. Clear? Clear, bache? So these are the 4 whorls. Okay, these are the 4 whorls. And let me tell you, students, this one is the outermost whorl. Okay, the calyx is the outermost whorl. And when you talk about the gynoecium, gynoecium is the innermost whorl. And do you know why? Do you know why? Because you know that it is the female part. Here it is having this pistil, or you can say that carpel. So what do we study here in this pistil and carpel? You know that this flat structure, it is stigma. This tube-like structure, style, right? Stigma, style, and the ovary. Okay, stigma, style, and the ovary. And here in this ovary, we will see the ovule is present. Okay, what is present here? The ovule is present. So this female part, you know that after fertilization, post-fertilization, you know that here what will you get? This ovule will form the seed. Clear, bache? So it is present within the ovary. So that is why in the case of angiosperms, we say that the seeds are covered. They are not naked because they are present. The ovule is present within the ovary. So after fertilization, this ovule will form the seed and it will be covered. So this is the female part which is going to form the seed. So it needs more protection, bache. That is why gynoecium is the innermost whorl. And when it comes to the outermost whorl, it is the calyx. Any doubt? It is the calyx which is the outermost whorl. Any doubt? Yes. So calyx, corolla, androecium, gynoecium, these are the 4 whorls, right? So calyx and corolla, they are the accessory whorls. They are the non-essential whorls. Their main function is of protection and to attract the insect. But when you talk about androecium and gynoecium, right, androecium and gynoecium, these are the essential whorls. These are the reproductive whorls. They are the one which will take part in the reproduction. Clear, bache? They will take part in the reproduction. So that's important. So as I said, when you are talking about a flower, especially a complete flower, okay, especially a complete flower. So when we say that flower is complete, it means it is having all the four whorls. Okay, when we say, Trisha, that a flower is complete, it means it is having all the 4 whorls. Okay, it is having all the 4 whorls: the calyx, the corolla, the androecium, and the gynoecium. And such flower will be, yes, such flower will be bisexual, right? Such flower will be bisexual. So for bisexual flower, this is going to be the symbol. Okay, this is going to be the symbol. So I hope you remember that trick, right? BMW, instead of BMW, make it BMH. B stands for bisexual, M stands for monoecious, H stands for hermaphrodite. Okay, B stands for bisexual, M stands for monoecious, and H stands for hermaphrodite. Clear, bache? H stands for hermaphrodite. Okay, so in your notes, this is what you can add. That these two are the non-essential whorls and here these two are the essential whorls. So essential one, why essential? Because they are the reproductive whorls. Okay, and non-essential, they are known as accessory whorls. So their main function is of protection and to attract the insects. Clear, bache? And to attract the insects. Fine? Fine. And secondly, see, now I just said that this flower is bisexual because it is having all the 4 whorls present. See, it is having all the 4 whorls present, so we will call it as a complete flower. Why am I calling it as bisexual? Because it is having male part as well, female part as well. In one flower, you have the stamen as well, you have the pistil as well. But sometimes you know that, what is the scenario? That these flowers, they are even the dioecious. Okay, they are what? They are dioecious. Dioecious means they are unisexual. So in that case, if it is a male flower, okay, so male flower means, if it is a male flower, it means it contains the stamen only. Okay, it contains the stamen only. So we will call it as staminate. Right, and if it is the female flower, it will be pistillate. Only female part is present here, so it will be pistillate. So such flowers are unisexual flowers. Staminate, pistillate, staminate, pistillate. Okay, done. Is it clear? And if there is any doubt, do let me know, please. Okay. So now we have to understand the pre-fertilization events. So calyx, corolla, very simple, bache. I am not going to explain that in detail in the chapter morphology, right? We talk about the sepals and the petals. We discussed that these sepals can be fused as well, right? They can be free as well, right? Remember, fused sepal, gamosepalous, polysepalous if it is free. So we have already discussed it there in the chapter morphology of the flowering plant. So today, what are we going to start first? We will start with what? We are going to start with the, yes, we are going to start with the pre-fertilization events. But before that, see, when you are talking about this stamen, it is given in NCERT, that's why I am telling you. See, in the case of stamen, this part, this lobe-like part, right? These two lobes, this is the anther and this filament. this filament is present here, and together they are making the stamen. So you know that this filament is attached to the thalamus. Yes or no? You know that this filament is attached to the thalamus. Yes or no? Tell me. The filament, it is attached to the thalamus. Yes or no? Yes or no? So this is a previous year question. Actually, it is the proximal part of the filament. Okay, it is the proximal part of the filament, and this one is the distal part. So proximal part of the filament is attached to the thalamus. This is what you need to remember, and it is given in NCERT, and it is a previous year question, that it is the proximal part of the filament which is attached to the thalamus. That's what you need to remember. Now let's read it quickly from the NCERT, and then we will start the pre-fertilization events. Okay, right? So in every lecture, bache, we will all I always start with these. I always start my explanation, and then we revise everything from the NCERT as well. So while attending the session, always keep your book with you, and whatever points are important, please highlight it, and you can even write down them on your book itself. Okay. So see, these are the general things that you know, that plants, they reproduce sexually, right? The myriads of flowers that we enjoy gazing at, and the scents and the perfume that we swoon over, the rich colors that attract us, all of them are the result of the sexual reproduction. So these are the general things, right? They are telling us that how this process is even important for us. You know that flowers, they are very beautiful. We use them in different ways, and even we extract the products from them. We even extract the perfumes from them. So this is what they have explained here. Now, when you talk about this flower, a fascinating organ. Actually, it is. Actually, it is an organ where the sexual reproduction is going to take place. We will get the seeds, and that seeds will help in the propagation of that particular plant, right? Okay, so human beings that have had an intimate relationship with the flowers since the time immemorial. Flowers are objects of aesthetic, ornamental, social, and religious and cultural value. I don't think that I need to explain it. You know about it, isn't it? You know about it. So here, this diagram is important. So here you can see the stigma, style, and the ovary, the pistil part, or you can say that the carpellus. Clear? And here you can see the bilobed anther and the stamen. Bilobed anther and the stamen, the platyls, and these are the sepals. So here this part is what? It is the thalamus. If stalk is present, that is the what? That is the pedicel. Clear, bache? Okay. Done. So that is given here. So to a biologist, flowers are the morphological and embryological marbles and the sites of the sexual reproduction. Right? They are the morphological and the embryological marbles and the sites of the sexual reproduction. So this is the part that we need to start now. The structure and the events here, right? So it will come under the pre-fertilization events. Okay, done, bache? Done? Any doubt? Sure? Sure? That's what we need to discuss. Fine. They think of floriculture is the study. It's the study of the flower, about their marketing and everything. Okay, that's very simple. Floriculture is very simple, bache. It is nothing. It is just the study of the growing flowers. How will they grow? They're about their cultivation and everything. That will come under the floriculture, right? So it is the study of that flowers. Okay. So now let's start the pre-fertilization event, the structure and the events here. Okay. So, let's relate it with pteridophytes first. Okay, that will be very good for your plant kingdom revision. See, in the case of pteridophytes, that's how we start. In the case of pteridophytes, we see that they are the first successful land plants. Isn't it? Like your ferns, right? When you are talking about the pteridophytes, means you are talking about the ferns. So you know that what are they? They are the first successful land plants. Now in the case of these pteridophytes, they are properly differentiated into true root, stem and leaves. Okay, in the case of pteridophytes, you will see true root, stem and leaves, isn't it? What do you have here? True root, stem and leaves. That's what we have here in the pteridophytes, right? That's what we have here in the pteridophytes, true root, stem and the leaves. Now in the case of pteridophytes, that's what we study, that some leaves, okay, some leaves, they will be modified. How will they get modified? Like these some leaves, they start bearing some structure in which spores will form, isn't it? These leaves, they will start bearing some structure, right? They will start bearing some structure in these structures the spore formation will be there. Like this is the leaf having sporangium. What is the meaning of sporangium, bache? The structure in which spore formation will be there. What is the meaning of sporangium? The structure in which spore formation will be there. I hope you know about it. Now, this particular leaf, because it is bearing a sporangium, we use the word sporophyll for it, right? We use the word sporophyll for it. Do you know about it? Tell me, do you know about it? But we are saying that let's start with the pteridophytes. No doubt we will relate it with the angiosperms. So in the case of pteridophytes, I'm saying that they are the first successful land plants and they have true root, stem and leaves. And when you talk about their leaves, some leaves will get modified. They will have the spore forming structures, right? They will have the spore forming structures, right? Like sporangium. Okay, sporangium, group of sporangium is there. There are many things, but I'm telling you the basics here. So we are calling it as sporophyll. What are we calling it? We are calling it as sporophyll. Now you know that after pteridophytes, we have the gymnosperms. After gymnosperms, we have the angiosperms. Obviously, they will be advanced, right? Gymnosperms and the angiosperms, they are more advanced than the pteridophytes. Okay, so now when you talk about the angiosperms, that is your flowering plants. When you talk about the angiosperms, your flowering plants, of course in their case also we will talk about the spore formation. We will discuss microsporogenesis and the megasporogenesis. So of course here also we will talk about these microsporophylls and the megasporophylls. So what will be the meaning of this microsporophyll? What is the meaning of microsporophyll? A leaf-like structure, right, having microsporangium in which microspores will form. What are that microspores? They are the one which will further form the male gametophyte. Are you getting it? Which will further form the male gametophyte. Is that clear? Is that clear? So in the case of angiosperms, the stamen is equivalent to, it is the stamen which is equivalent to microsporophyll. Fine, that's why I'm explaining you this. It is equivalent to microsporophyll. It is the structure in which the microspores will form, right? So it is equivalent to microsporophylls. And the pistil or the carpel, it is equivalent to megasporophyll. That's important, bache. Okay, so it is equivalent to megasporophyll. Is it clear? It is equivalent to megasporophyll, right? These 2 points are important. Fine. These 2 points are important. Done, bache? Done? Sure? So stamen equivalent to microsporophyll, pistil equivalent to megasporophyll. So now, bache, when you talk about the flower, so how will it be decided that flower is going to form? Of course, the hormonal changes will be there, right? In the case of plants, we know about the growth regulators. We know that growth regulators, the one which are going to control the growth, they are present in the case of a flower, right? So because of that hormones, because of that changes, because of that physiological changes, what is going to happen? Shoot apical meristem will form floral meristem, and that floral meristem will form floral primordium. It will form the structure which is going to form the flower, bache, right? So now in the case of this flower, right, we are going to start with the male reproductive organ. How are we going to start with? We are going to start with the male reproductive wall, and you know that what is it? It is the stamen. It is the andrecium. Clear, bache? It is the stamen. It is the andrecium. Fine. Are you getting it? Right. So male reproductive organ is andrecium. Male reproductive organ is andrecium, having whorls of stamen. And bache, these stamen, their number and length. Number of stamens and length of filaments. It varies. Number of stamens and length of filament. It varies from species to species. Remember this always. Okay, it varies from species to species. In different species, you will see the number of stamen is different, even their length is variable. And sometimes in a flower, right, you will see that in the whorl of stamens, some stamens are long, some are short as well. So these things, they are variable. Clear, bache? They are variable. Done? Done. So now you know that two parts are there in the stamen: bilobed anther and a stalk-like structure, which is the filament. Right? This anther, it is lobed, it is kind of condensed structure, and this filament is a stalk-like structure. Clear, bache? So this filament, the proximal part of this filament, fine, the proximal part of this filament is attached to the thalamus. Write down. Proximal part of filament is attached to thalamus. Clear, bache? It is attached to thalamus. Done? It is the proximal part of the filament which is attached to the thalamus. Clear, bache? Clear, clear, clear. So when it comes to the attachment of the anther and filament, okay, when it comes to the attachment of the anther and filament, they have the connective tissue here. What type of tissue is there? They have the connective tissue here. So now mainly we need to understand about this anther, about its internal section as well. Fine, bache? Done? Sure? Sure? So now, bache, when we talk about this anther in a typical angiosperm, like we consider the example of Capsella, shepherd's purse, right? That is a typical angiosperm that is considered to study all that things. So when you talk about this anther, what is it? It's bilobed. What is it? It's bilobed. It's dithecous. It's tetrasporangiate. It's bilobed. It's dithecous. It's tetrasporangiate. Okay, these are the important words. Bilobed, dithecous, tetrasporangiate. I'm repeating it again. Bilobed, dithecous, tetrasporangiate. I'm not saying that in all the angiosperms this is what you are going to see. No, nothing like that. Like if I'll give you the example of family Malvaceae and you will see the differences there. But mainly in a typical angiosperm, mostly what will you see? That the anther is having two lobes. Right, anther is having two lobes. Now when you talk about each lobe here, these two lobes are having two theca. Focus here. Okay, we are saying that we have the two lobes here, and now each lobe is having two theca. Right, two spaces. Like let's say you are saying that you have two flats. Okay, you are saying that you have two flats and each flat is having two rooms. Fine, it's more like that. Let's say you have two flats and each flat is having two rooms. This is what you can understand. Like that is how you can understand. So why am I saying that anther is bilobed? Because two lobes are present here. And why do we use the word dithecous? It means each lobe consists two theca. Fine. So theca is nothing, it's a kind of space there, right? It's a kind of space, and when the pollen grain it will mature, this theca will form microsporangium. Okay, this theca is going to form microsporangium. Are you getting it? Are you getting it? Yes? Yes or no? So this point is important. Again, I'm repeating, bache, that there is, just let's say you have one pen, this single pen is having this single pen is having two refills, right? The single pen is having two refills. So you can understand it in this way. So ultimately two refills are present there. So if I will take the section here, okay, if I will take the section here, the cross section here, so this is what you will see, that this is the outer body of the pen, and here you will see the two refills, the cross section of that two refills. Same is the case here. Okay, two lobes, and each lobe is having two theca. Two lobes, each lobe is having two theca, and bache, there is a longitudinal groove. Okay, what else is there? There is a longitudinal groove which will run lengthwise and divide this theca. Okay, what is there? There is a longitudinal groove which will run lengthwise and it will divide it. Done? Something like this. Something like this. So there will be the longitudinal groove which will run lengthwise and it will divide these two theca. Clear, bache? Clear? So that's how you have to remember this. And again, always remember the anther and filament, they are also attached with the help of connective tissue. Now, when you look at this particular structure, as I said, this theca, right? Slowly, you know that your pollen grain will, right? Pollen grain will mature, isn't it? The pollen grain will mature. So obviously here some changes are going to take place. Of course, 110% some changes will occur here, right? So now these theca, this theca will form the microsporangium. What will it form? It will form microsporangium. So now, bache, micro means small, sporangium, spore bearing structure, right? It's a structure in which microspores will form. What is the meaning of microsporangium? The meaning of microsporangium is it's a spore, it's a microspore forming structure. You know that in nature, the male gametes are small in size, female gametes are large in size. This is what we know, that when it comes to the male gametes, they are more in number, they are small in size. Female gametes less in number, but they are larger in size. So that's the case here. So when we are saying microsporangium, the structure in which microspores will form. Clear? So now this theca will grow. When the pollen grain will grow, obviously some changes will occur here in this theca. Right? Some changes will occur here in this theca. So this theca, this part here, this in not just the theca is a kind of space present here. Right? Theca is a kind of space present here. So this entire portion, this structure here, right? This complete structure here, we will use the word microsporangium for it. Right? Microsporangium for it. So if I talk about the typical anther, so you know that it's bilobed and dithecous. So when you look at it, right? Just look at it. Two lobes are there. Yes, lobe number 1, lobe number 2. Each lobe is having two theca. That's why dithecous. Now, overall, when you look at this anther, don't you think that these four sides, they are having 4 microsporangium? They are having 4 microsporangium, right? So that's why we use the word tetra. Tetra means 4, that it is tetrasporangiate. It's tetrasporangiate. Clear? What's that? It's tetrasporangiate. Done. And here in between, right, it is just the connective tissue. It is the connective tissue. And here in between, we will also see the vascular strand. Is it clear or not? Yes, bache, is it clear or not? So microsporangia, it will develop further. So first of all, anther is having 4 microsporangia located at the corners. So they will further develop, right? They will further develop these microsporangium, and these microsporangium are going to form the pollen sacs. Okay, so theca is just that longitudinal, that space present in between. That's how you can remember. So this complete structure with all that layers, these 4 corners, they are forming 4 microsporangium, and then this microsporangium will further grow and it is going to form the pollen sac. So somewhere, right, they all are same. जैसे theca है, 2 theca, that spaces are there, the theca will further grow, and all that layers, everything, right? This typical portion here, we are using the word microsporangium, it will further grow, it is forming the pollen sac. And here you will see that these pollen sacs, they will grow lengthwise, longitudinally they will grow in that pollen sac. Can you imagine that? Longitudinally they will grow in that pollen sac. Are you getting it? So in my case, if I have to visualize it, I always think of a pen having 2 refills, right? Right? Or you can even consider a pen, multi-pen, which is having 4 refills. 4 refills are there. So 4 different compartments are there. Okay. Okay. So that is how you can remember that. Fine. Fine, bache. Yes or no? Done. Done. So as I said that we have the exception as well, that in Malvaceae, okay, so anther is, it's bilobed, but it is monothecous. Now as here we are saying it's bilobed but dithecous, but here it will be bilobed but it will be monothecous. Like there will be the, let's say, only 1 theca will be there. Only 1 theca will be there. Here you have 2 theca in each lobe. Here you have 1 theca in each lobe. So anther is bilobed, right? Monothecous. So obviously it will be bisporangiate. So this exception is important. Clear, bache? This exception is important. So in Malvaceae, the anthers, they are monothecous and bisporangiate. They are monothecous and bisporangiate. Clear, bache? Sure. So see this. So before the actual flower is seen on a plant, the decision that plant is going to form flower has taken place. Okay. Several hormones and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Right. Right. So before we see that flower on a plant, right, plant has done all the possible changes for its formation. That's what they are saying. So you know that inflorescence are formed which bear the floral buds and then the flowers. What is inflorescence? Right? Group of flowers. Inflorescence. Okay. So you know that we have that, you know, all that floral axis and everything. Now I don't think that I need to explain it. So inflorescence, hope you remember the group of flowers, or you can say that the arrangement of flowers on the floral axis is also the inflorescence. Clear, bache? So male and female reproductive structures are there in a flower. Androecium and gynoecium will differentiate and develop. Okay, androecium and gynoecium, they will differentiate and develop, and that's the diagram that I'm talking about. So here you people can see, right, 2 lobes, and here this is the filament. And as I said, anther and filament, they are connected together by a connective tissue. And see, this is the diagram here. Can you see this? Now we will draw it. So here you can see each lobe is having 2 theca and a longitudinal groove. Okay, it is separating it. So it is also forming the line of dehiscence. So what is the meaning of the line of dehiscence? Right, it is the part from where the anther will rupture. Once mature, it will rupture from this line of dehiscence and all the pollen grains, they will be dispersed. Clear, bache? Clear? So we are talking about the stamen, which is equivalent to the microsporophyll, and we'll be talking about microsporangium and the pollen grain here. Done, bache? Microsporangium and the pollen grain here. So see, let's revise it from NCERT. That typical stamen, the long and cylinder stalk, the filament, and terminal end is the anther. So yes, they can ask you, this is the terminal part here, distal part here, and this one is the proximal part here. Clear? This one is the proximal part here. Okay. Okay, so proximal end is attached to the thalamus or the petal of the flower. MCQ. Fine, MCQ. Done. So, bache, if this stamen, if it's stalk, if it is attached to the petal, fine. If it is attached to the petal, do you know how do we show it? How do we show it? This. It is showing the adhesion. Okay, in morphology, that's what we discussed. It is showing the adhesion, that your A is attached to the C. A is attached to the C. C means petal, A means androecium. So your stamen, your stamen is attached to the petal. Clear, bache? It is showing the adhesion. Fine. So the number and length of the stamens, they are variable in flowers. I told you already. Clear? So if you were to collect a stamen, each from 10 flowers, and arrange them on a slide, you would be able to appreciate the variations. Okay, this is just an experiment that you want to, if you want to do, you guys can. Okay, so a typical angiosperm, bilobed, dithecous, right, and a longitudinal groove runs lengthwise, separating the theca. Clear, bache? Separating the theca. Done. So bilobed nature of anther is very distinct in the transverse section, and anther is a four-sided. It's a tetragonal structure, bache, having 4 microsporangia located at the each corner of each lobe. Done. Each corner of each lobe. So that's what we need to discuss now. Now we will talk about the structure of the microsporangium, the most important topic. Okay, that's what we are going to discuss now, the structure of the microsporangium. Clear, bache? So this is what you can mark, that your microsporangia, it will develop further and it will form the pollen sac, and these pollen sacs, they extend longitudinally all through the length and are packed with the pollen grains. Fine. Fine. So that's our topic that we need to start now. It is the structure of microsporangium. Right, it is the structure of the microsporangium. Clear? Fine. It is the structure of the microsporangium. Now see, typically that's what we are saying, bache. Typically, that's what we are saying, that when we look at the transverse section here, the TS here, right, it looks like a tetragonal structure, and each corner here will have that microsporangium, like this part, this part, this part, and this part. And once again, I'm repeating, bache, here you will find the connective, and in between there will be a vascular strap. Okay, so here in between that cells that are present, okay, they are basically the parenchymatous cells. What are they? They are basically the parenchymatous cells. Please let me know what type of cells they are. They are the parenchymatous cells. Okay, parenchymatous cells. Now we need to focus on this particular part. Can you see this? This particular part. This is what we are going to study now. Just say here, the outermost layer. Let's start with the outermost layer. This outermost layer is epidermis. What is it? We are going to start with the outermost layer here, right? They say here it is just the general outline, but we have the different layers present here. So the outermost layer is what? It is epidermis. And whenever we talk about the epidermis, you know the role of epidermis, you know the main function of epidermis, that it is meant for the protection. Okay, it is meant for the protection here. So epidermis, outermost layer, single celled it is, and it is the protective layer. Okay, it is outermost layer, single celled, uniserrated it is, and it is protective in function. What is it? It is protective in function. Now, bache, just below this, right, just below this epidermis, the another layer which is present is your endothecium. What is it? It is your endothecium. Wait. It is your endothecium. Just below this endothecium, we have 2 to 3. First of all, understand the sequence of layers. Okay, what do you have? You have 2 to 3 middle layers. Right, you have 2 to 3 middle layers. Clear, bache? I will explain you the cell structure and everything. Right now, I am telling you the sequence. Outermost layer epidermis. Below epidermis, there will be endothecium. Below endothecium, there will be 2 to 3 middle layers. Right, and then the innermost layer will be tapetum. What is it? It is a tapetum, and this tapetum is covering the sporogenous tissue here. Clear? This tapetum is covering the sporogenous tissue here. Are you getting it? So this is the structure of a microsporangium. This part having different layers. Okay, this part is having different layers. So the outermost, as I said, outermost is epidermis, then endothelium, then middle layers, and the innermost will be tapetum. And this tapetum is covering this portion, and it is nothing. It is the sporogenous tissue, the tissue which will form the spore. Okay, it is the sporogenous tissue, the tissue which is forming the spore. Clear, bache? The tissue which is forming the spore. So now we need to understand about it. So here, bache, the layers are very important. Clear? What is very important here? The layers are very important. Clear? So outermost epidermis. Endothelium. Done. And then the next to it is tapetum. Now I will tell you about the features, right? That which layer is having what type of cells. That's what we need to discuss now. So this part is the sporogenous part. Okay, this part is the sporogenous part. So again, epidermis, endothelium, bache, the sequence of layers. It is very important. Middle layer, this white part. Tapetum. Basically, it's tapetum. Okay, so these are the 4 layers. Right? These are the 4 layers. So now let's start it. This is important. Please make the notes. Outermost, unilayered or protective in function. Clear, bache? Protective in function. Next to it is endothelium. Next to it is what? It is endothelium. Do you know this endothelium is also known as the fibrous layer? Are you aware of it? When you talk about this endothelium is also known as the fibrous layer. But here you will see some thickenings. Do you know that? In this particular layer, what will you see? You are going to see some thickenings here, right? No doubt it's single layered, right? Even it is single layered. But here, like let's say, so you know that it is just below the epidermis. It is also single layered. But here when you talk about the cells, like let's say, these are the cells. So here you will see, bache, listen to me carefully, then I will write down, right? Here, the outermost layer of the cell, outermost layer is going to be thin walled, right? What am I saying? That no doubt this endothelium, it is the fibrous layer. It is just below the epidermis. And here I'm saying that here the outer layer is going to be thin. But when you talk about its inner layer, when you talk about its inner layers, see these one, inner and even the radial wall, they have some alpha cellulose thickenings. Okay, they have alpha cellulose thickenings, right? They are comparatively thick. Right? Out the inner and the radial walls, they are comparatively thick. Why? Because they have the cellulose fibers. What do they have? They have the cellulose fibers. Clear, bache? Clear, bache? And when you talk about the radial walls here, right? Radial walls also contain callose bands. Right? They also contain callose bands. So callose is also a polysaccharide. Callose is what, bache? Callose is also a polysaccharide. Done. So endothecium, single layered, outer wall of cell layer is thin, right, is thin. Inner and radial walls contain alpha cellulose, fibrils. Right. Next point is radial walls also contain callose bands. Is it clear? Radial walls also contain callose bands. Clear, bache? Clear, bache? Yes. Now, these callose bands, see, this is the diagram. Okay. So, see, this is from NCERT. The rough structure, the outline, the layers, and this is the mature anther. Okay, it is the mature anther, and in mature anther you will see middle layers are not there. See, this is the mature anther. So, bache, this part here, basically this part here is known as stomium. This part here, it is known as stomium. What is it? It is stomium. Yes, bacho, what is it? This part here, what is it? It is the stomium. Now, why stomium? Why stomium? Because stomium, see here, endothecium, cells of endothecium are without callose bands, right? This part is what? It is the stomium, right? It is the stomium, bache, and here you will see what will you see here? That here these cells of endothecium, they are without callose and fibrous thickenings. Fibrous thickenings means without that cellulose fibrils. Okay. Okay. So this part, because it is not so thick, so it is helping in the dehiscence. How is it helping, bache? It is helping in the dehiscence. Are you getting it? How is it helping? It is helping in the dehiscence. Done? Done, done, done. Sure. So this is the stomium where callose band and alpha cellulose fibers. Actually, it is the fibers, not the, right? So alpha cellulose fibers are absent here and the callose bands are absent here. So this, from here, the dehiscence of anther will occur. Is that clear? Stomium clear? Yes. Stomium clear? Sure. That's important. Done. So radial walls also contain callose bands. Are you getting it? Radial walls also contain callose bands. Clear, bache? So epidermis, then endothecium. Okay. So endothecium is hygroscopic in nature. Do you know the meaning of being hygroscopic? That it is hygroscopic in nature. Bache, it means it is going to absorb the moisture from the surrounding. Fine, it is going to absorb the moisture from the surrounding. I will explain you the mechanism of the dehiscence as well. So imagine it will absorb the moisture from the surrounding, so it will help in that dehiscence. Clear? Clear, bache? Right? The pressure will also increase on the cells. There will be the bursting then. Fine, there will be the bursting then. So endothecium hygroscopic in nature due to the presence of this. Why? Because of the presence of these fibers. Okay, so endothecium become hygroscopic in nature due to the fibrous thickenings. Because of these thickenings, it will become hygroscopic in nature and then help in dehiscence. Done? Any doubt? Yes? Any doubt here? Sure? Sure, sure, sure. Done, bache? Okay, so next to it, as I said, 2 to 3 layers, 2 to 3 layered middle layer is present. Basically, you can simply say middle layer is present below the endothecium. And when you talk about middle layer, it is multi-serrated. What is the meaning of multi-serrated? That it is having many layers. So it is 2 to 3 layered thick. What is it? It is 2 to 3 layered thick. It is 2 to 3 layered thick and made up of parenchymatous cells. It is made up of parenchymatous cells. Now, bache, I hope you know the meaning of parenchymatous cells. Living cells they are. They are the one in which food will get stored. Okay, they are the one in which food will get stored. So 2 to 3 layered thick made up of parenchymatous cells. Food is stored here. And do you know that this particular layer, it is short-lived? This particular layer, it is short-lived. For short-lived, we use the word ephemeral. Right, it is short-lived, so we use the word ephemeral. Basically, this layer will not be there in the mature anther. Why? Because it is mature now, it doesn't need the food, it is already having everything, right? It has utilized that particular food for its nutrition, for its growth and development there. So when you will see the mature anther, you are not going to find the middle layers there. Okay, we are not going to find the middle layers there. So it is 2 to 3 cell layered thick, right, made up of parenchymatous cells. The food is going to be stored there, and as I said, the middle layer is ephemeral, that is short lived, and it is absent in mature anther. Okay, it is short lived and absent in mature anther. Okay, so these 3 outermost layers, bache, they help in the dehiscence of the anther mainly. Okay, these 3 outermost layer, their main function is of the dehiscence of the anther, and of course they are also providing the protection. Clear? Clear? So write down. So these 3 outermost layers help in protection and dehiscence of anther. Clear, bache? And dehiscence of anther. Done? Done? So the next to it is what? Next to it is tapetum. Now this is very important. Can you just see the diagram, bache? Here in this diagram, I made 2 dots because actually these 2 dots are representing, right? They are representing the binucleated state. When you talk about the tapetum, it is nutritive and the secretory layer. What type of layer it is? It is nutritive as well as secretory. Nutritive means it will provide nourishment to the sporogenous tissue. It will provide nourishment to these spores here, right? And secretory, obviously it will release the things which will help in, which will ultimately help somewhere. It is going to help in that microsporogenesis and all. So when you look at the tapetum, it is not uninucleated. The cells here, they are multinucleated, and you can say that binucleated they are. Why? Because here you will see endomitosis and the polyploidy. I will explain it. Wait. So this tapetum, it will also release the sporopollenins for making the enzymes of the pollen grain. It will also release the callase enzyme, which will also help in dissolving the callose. I will tell you about that too, right? And moreover, it is providing the nourishment to this sporogenous part. It will take all the food, all the nourishment from the middle layer, and then it will give it to it. Okay, so the function of tapetum, they are very, very important. Okay, the functions of tapetum, they are very, very important, and this is what now we are going to discuss. Okay, so innermost nutritive and secretory layer it is. Fine. Innermost nutritive and secretory layer, and it is also, bache, 1 cell layered thick, means it is also uniserrated. Fine, it is also uniserrated. Clear, bache? Tapetum. So it is the innermost nutritive and secretory layered, which is 1 cell layered thick. What is it? It is 1 cell layered thick. Are you getting it? Are you getting it? So, bache, cells contain dense cytoplasm. See, whenever you will see now that particular layer is secretory, you will see the cytoplasm is going to be dense there. Okay, the cytoplasm is going to be dense there, and as I said, bache, it is having more than one nucleus. Okay, it is having more than one nucleus. Clear? Clear? So it is important to remember that it is having more than one nucleus. Then it can be multinucleated, it can be binucleated or multinucleated. Now, bache, initially the cells of tapetum, they are diploid. Initially they are diploid, but then here na endomitosis will occur. So endomitosis and endomitosis will occur here. Do you know the meaning of endomitosis? Yes. Anyone here in the class? Do you know the meaning of endomitosis? What is the meaning of endomitosis? Anyone? Just give it a try. Tell me. What is the meaning of endomitosis? Tell me. Mitosis is there, but the word is endomitosis. Endomitosis is the word. Endo means inside. So actually here what is happening? See, this is a cell and here the nucleus is diploid initially, but then the endomitosis will occur. But endomitosis is also known as endoreplication. Here the nuclear genome, here the genetic material is dividing. The genetic material is dividing, but you will not see two cells here. Genetic material is dividing, right? Genetic material is dividing, but there will be no further nuclear division there, right? There will be no further nuclear division there. Two different cells will not form. It is just the genetic material which will get replicated. So obviously if initially it was diploid, then of course what is going to happen, bache? What is going to happen? The genetic material will get doubled here. So obviously it will not remain diploid. Slowly it will become polyploid. Right, whenever we talk about the tapetum, the cells, the nucleus here, we use the word that the cells, it is multinucleated and it is polyploid. It is not diploid. We see the polyploidy here, right? We see the polyploidy here. Why? As I said, what is going to happen here, bache? The endomitosis will occur, right? Endomitosis will occur. Okay, so initially endomitosis will be there and then nuclear division. So initially it is diploid, but because of endomitosis, as I said, your nucleus is not going to divide initially. Your nucleus is not going to divide initially. It's just the genetic material which will get replicated, which will get doubled. Okay, so obviously if genetic material will get doubled and you are not getting two daughter cells, so of course here you will see the polyploidy. Okay, you will see the polyploidy, but after some time there will be the nuclear division as well. So you will see, right, more than one nucleus. You will see more than one nucleus plus that nucleus is what? It is polyploid. It is polyploid. Are you getting it? It is polyploid. That is the meaning here. So any doubt? Yes, any doubt? So endomitosis and nuclear division will result in the polyploid cells here. Sure? Sure. Done? Sure, bache? Okay, so before dehiscence, that's important. Okay, so before degeneration of tapetum, it releases Yubish bodies. This is important, please write down. It releases Yubish bodies. Okay, so what is this Yubish bodies? Just in some books, it is simply written as sporopollenin. right? It is simply written as sporopollen. Actually, bache, what is happening? There are some structures, right? Like there are some pre-ubish bodies, right? They will be covered with the sporopollen. So actually, the sporopollen is forming the ubish body. What is it? It is the sporopollen. Sporopollen is an organic substance, right? This sporopollen, it is going to form the outermost layer of the pollen grain, that is exine. What is going to happen, bache? Before degeneration of tapetum will form the ubish body. As I said, what is the ubish body? You will see that there will be some secretions which are covered with sporopollen. For that, you are going to use the words ubish bodies. Now these ubish bodies, they will be, before degeneration of tapetum, they will be released in the pollen sac. Where will they release? They will be released in the pollen sac before the degeneration of the tapetum. And the sporopollen will do for what? It will form the outermost layer of pollen grain. Okay, outermost layer of pollen grain, and that outermost layer of pollen grain is known as exine. Okay, it is known as exine. So that's how it is helping. Fine, bache, that's how it is helping. So the function of tapetum, they are very important. Clear? The functions of tapetum, they are very important. So this is also one of the functions, the formation of ubish body or the secretion of sporopollen, which is the hardest organic substance known. Clear? Which is the hardest organic substance known. And moreover, I told you already that this tapetum, it will nourish the microspores, right? It will help in the nutrition of these pollen mother cells also, and in the nutrition of these developing pollen grains. Okay, bache? Clear? Sure. And moreover, this tapetum, it releases an enzyme callase. So please write down the function that firstly it will provide nourishment to the pollen mother cells, to the developing pollen grains. It releases an enzyme callase. I will tell you the function of callase as well. Clear? It is going to release the enzyme callase. Which enzyme will be released by this? Callase. It's callase. Okay? Okay. Done. And even you know that this pollen grains, right? So these tapetum, they will also form the pollen kit substances. Fine. They will also form the pollen kit substances. Done, bache? Okay. So now here, when you look at this sporogenous tissue, right, when you look at the sporogenous tissue, no doubt all the cells are diploid here. Fine. All the cells are what? They are diploid here. What are they, bache? These cells are diploid here. Now you know that in the case of males, male gametes, their number is more, isn't it? Their number is more. So when you look at that sporogenous tissue, let's say this is the sporogenous tissue here. And as I said, the sporogenous tissue is covered with a layer. And that layer is what? It is the tapetum, right? Which is going to provide the nourishment here. So it's a spore forming. What is it? It's a spore forming tissue. What is it? It's a spore forming tissue, and that's why I'm using the word sporogenous tissue here. Now, bache, each cell present here in this tissue, it can form the pollen grains. Now let's pick one cell only. Of course, it is going to be diploid, right? So let's pick one cell only. Of course, it is going to be what? It is going to be diploid. So this cell is your PMC or MMC. Now, what is the meaning of PMC and MMC, ma'am? It's pollen mother cell, right? It is pollen mother cell, and you can also call it as microspore mother cell. Okay, so each cell of the sporogenous tissue is having the potential to form the pollen grains, right? And let's pick up one cell here. Let's pick up one cell here. We are calling it as pollen mother cell or the microspore mother cell. It is diploid in nature. And bache, I always mention this thing in my lectures that when I'm saying mother cell means it will undergo meiosis. It will undergo meiosis. What is going to happen here, Yasir? It will undergo meiosis. So this is your diploid pollen mother cell. So now when you talk about the meiosis, you know that meiosis is the reductional division, isn't it? Meiosis is what? It is the reductional division. Soumyadeep, what is meiosis? It is the reductional division. So it is the reductional division means it will reduce into, it is the reductional division, it is going to reduce the chromosome number to half. Let's say here you have 10 chromosomes. So after meiosis 1, you will have 2 daughter cells having 5 chromosome. And after that again, you will have meiosis 2. You know that meiosis used to occur in 2 rounds. There will be 2 rounds of nuclear division and cell division. So after meiosis, we used to get 4 daughter cells having haploid number of chromosomes, right? Having haploid number of chromosomes. So 10 after meiosis 1, 2 cells having 5 chromosomes. After meiosis 2, 4 daughter cells, 5 chromosomes. Clear, bache? Clear, bache? That's what meiosis is. So now this pollen mother cell, as I said, it will undergo meiosis 1. You will get 2 haploid microspores. Now they are what? They are the microspores. And after that meiosis 1, there will be meiosis 2, and you will get 4 haploid microspores. What will you get? You will get 4 haploid microspores, right? So this is microsporogenesis. This is microsporogenesis, right? What is the meaning of microsporogenesis? Formation of, genesis means formation of microspores. What is it? It is the formation of microspores. So at the end, you will get 4 haploid microspores. And bache, these microspores can be arranged differently. Like let's say if they are arranged like this, it is isobilateral arrangement. Fine. And it is very common in the case of monocots. And they can also arrange like this, tetrahedral arrangement, right? What type of arrangement it is? Tetrahedral. It is very common in the case of dicots. Fine. It is very common in the case of dicots. Clear, bache? So initially these microspores, they all are joined together. They are not separated initially. Fine. They are not separated initially. They are joined. Clear? And they are joined with the callose. Callose is a polysaccharide. Callose is a polysaccharide. So that callose is placing them all together. Initially, these microspores are not separated. They are together, right? And I'm saying that these microspores can be arranged in a different ways. So if it is isobilateral, it is in the case of monocots. Tetrahedral in the case of dicots. Tetrahedral. So it is looking like that 3 microspores are there. No, one is behind. Just a national emblem is there, no? Right? Right? So you will see na 4. Basically, so here also in tetrahedral, 4 microspores are arranged. Clear? So how are they attached? They are attached with the callose. Callose is what? Callose is a polysaccharide. Okay. And moreover, this type of arrangement, it is linear. This type of arrangement, it is linear. And you will even see some T-shaped arrangement of microspores as well. So microspores can be arranged in a different manner. Clear, bache? They can be arranged in a different manner. Done? Done. Sure? Sure, bache? So linear, T-shaped, anything can be there. It can be decussate also. You also use the word decussate. So microspores are arranged in different ways. Fine. So this is the microsporogenesis. What is it? It is the microsporogenesis. And then moreover, bache, right? It is also explained in this way that there will be the primary parietal cell, that cells will divide. Archisporial cell will be there, that will divide. Primary parietal cell, sporogenous cells will be there. Sporogenous cells will divide further. They will form the sporogenous tissue. So it is also given like that. Like when you talk about the sporogenous tissue, obviously here some cells were there, archisporial cells. That archisporial cells, they divide mitotically, they increase their number. They form that primary parietal cell. They form that, what we call it, the sporogenous cells. So that is how we discuss it. That initially it used to be archisporial cell which divided, which is going to form parietal cell and sporogenous cell. Parietal cells will further divide mitotically. They will form the layers, epidermis, endothelium, middle layers. Sporogenous cells, it will divide mitotically, it will increase its cell number, it will form sporogenous tissue. But because in NCERT it is explained in a simple way, that's why I'm telling you like this, that ultimately this is a sporogenous cell. Each cell is having the potential to form pollen grains. So you will pick up one cell. We are calling it as pollen mother cell or microspore mother cell. When I'm using the word mother cell, means it will undergo meiosis. After meiosis 1, 2 haploid microspore. After meiosis 2, 4 haploid microspore. So that is how they are arranged, right? And they are attached together with the help of callus. And now I told you now that tapetum, it used to release the enzyme callase. I just said na, tapetum, it releases the enzyme callase. So this callase will act on this callus. This callase will digest this callus, and all the haploid microspores, they'll be free. Clear, bache? They'll be free. Done. So rest. Okay, so rest we will discuss in the next class, where we will talk about the development of male gametophyte. We will see that how these microspores, then they will be covered with entine exine. We will talk about the pollen grains, and then, right, we will discuss it in the next class, because after now I have a class in Avengers batch also. So that's what we are. So today, this is what we are going to discuss. Okay? So what you have to do, you need to revise it from NCERT. Fine, just read it from NCERT and revise it from NCERT. So we have covered the microsporogenesis. Clear, bache? We have covered the microsporogenesis, and as I said, be the part of this batch, right? This is one of the best batches that you guys can ever have. And moreover, students, here when you see this, just a minute. So yes, right? So in the description box, all the links are there, and moreover, in the video section, if you will check the video section, you will see that I have posted one video on microsporogenesis and megasporogenesis. You can use that video, because that is the topic we will cover in next class. Just check that video. It is, I think it is for 19 to 20 minutes, right? Increase the speed, listen to that topic, right? And in next class, when we will do it, it will be a very good revision. Fine. So now you know what you have to do. You just need to write, you have to put one comment, and you have to tell me that you like the session or not. Okay, and which chapter you want to start next. Fine, which chapter you want to start next, and you like the session or not. This is what you need to tell me in the comment section. So I will wait for your comments. Fine, the PDF will be provided to you, right? You guys can join our official Telegram group, Unacademy NEET English, and I will share the PDF there. Fine. So thank you so much. So see you all tomorrow. Till then, bye-bye. Take care.